No fish has ever been found in the deepest 25% of the ocean. Not for lack of trying, life there might simply be biologically impossible. And yet, in almost every other hostile corner of the deep, evolution found a way. Take the zombie worm, feasting on the dead and dissolving bone with acid, or the Antarctic sea spider, swelling to 500 times the size of any relative. Life adapted again and again to conditions that should have made it impossible. This is the story of 4 enormous challenges to survival in the deep sea, and how life answered each one, and why, at a certain depth, it finally couldn't. The deeper you go, the less food there is. For creatures of the abyss, this leaves 2 options: eat less or get inventive. The Cuvier's beaked whale goes to extraordinary lengths to eat well. It holds the record for the deepest and longest dive of any mammal, plunging nearly 2 miles down on a single breath. At this depth, the ocean is absolute black. Vision is useless, so the whale navigates by echolocation, firing bursts of high-frequency clicks that bounce back and build an acoustic map of the water ahead. When those signals return from a squid, there's nowhere for it to hide. The whale snaps its beak open, creates a pocket of suction, and draws the squid straight in. Clean, efficient, and devastating. But most deep-sea creatures aren't apex predators. For them, food arrives in 3 main forms. The first is marine snow, a perpetual drift of organic material falling from the surface, dead zooplankton, animal waste, and fragments of phytoplankton. It descends slowly through the water column, feeding whatever it passes on the way down. Around 815 million tons settles on the ocean floor every year. That sounds generous until you spread it across the global seafloor, where it amounts to just 2 ounces of carbon per square yard. Enough to survive on, but only just. Nearly 1.25 miles further down, the vampire squid has built an entire existence around this falling feast. Long, sticky filaments hang between its arms. The squid extends them into the water and simply waits. Particles accumulate. When enough has gathered, it sweeps them in, wraps the catch in mucus, and swallows it whole. No pursuit, no struggle, just patience, and a system so efficient the vampire squid can survive on marine snow alone. Deeper still, lavaceans take a different approach. These small, tadpole-shaped animals build a mucus house around themselves, an elaborate filter that catches particles as tiny as bacteria. When it clogs, they abandon it. The discarded house drifts on as marine snow, feeding whatever lives further below. Not all marine snow reaches the creatures waiting in the middle depths. Much of it continues all the way down and settles on the ocean floor, with nutrients that sparse and spread that thin, moving around to find food costs energy you can barely afford. The gummy squirrel, a vivid yellow sea cucumber, has solved this elegantly. More than 3 miles down, it crawls slowly across the abyssal mud on tube feet, sifting through the sediment with specialized feeding palps and extracting the freshest deposits. When the supply runs thin, it doesn't trudge on. It waits for a current, extends its tail like a sail, and lets the ocean carry it to the next feeding patch. The second energy source powers some of the most biodiverse places in the deep ocean, and it does so without a single ray of sunlight. Nearly 2.5 miles down along the mid-Atlantic ridge, hydrothermal vents crack open the seafloor. Chimney-like structures blast superheated, mineral-rich water at 572 degrees Fahrenheit into the cold sea. The pressure keeps it from boiling, so it erupts instead as dark, billowing plumes. These vent fields can support over 300 species: anemones, shrimp, crabs, mussels, clams, and the iconic giant tube worm. At the base of every vent ecosystem are chemosynthetic bacteria. They don't need sunlight. They extract energy from the chemicals the vents release: hydrogen sulfide, methane, ammonia, and use it to build organic molecules. Think of it as photosynthesis running on a different fuel. Same result, completely different power source. The Pompeii worm takes this to an extreme. It bores its tube directly into the chimney walls. Its tail sits in water reaching 221 degrees Fahrenheit. Its head pokes out into cooler water at the other end. Across its back grows a thick mat of bacteria that absorbs and redistributes heat, a living insulating layer. The worm feeds the bacteria with mucus. The bacteria keep the worm from cooking. It is one of the most heat-resistant animals on the planet. The third energy source is the rarest and the most dramatic. After a long life crossing the open ocean, a bowhead whale takes its final breath and begins to sink. The descent takes hours, drifting slowly down past the marine snow, past the dark. It settles at last onto the soft floor of the abyssal plain, more than 2.5 miles down. This is one of the most barren environments on Earth. Organic matter here is so scarce that the arrival of a single whale carcass will sustain the surrounding ecosystem for decades. Rat-tail fish and hagfish arrive first, drawn from miles away by the scent of decay. They strip the flesh over weeks. What they leave is the skeleton. Whalebone contains roughly 200 times the nutrients of the surrounding seafloor. This is what the zombie worm has been waiting for. Its larvae arrive on ocean currents and settle onto the exposed bone, where the females transform into tube worms. One end develops into a fan-shaped structure that draws oxygen from the surrounding water. The other grows into a dense root system that bores deep into the bone itself. These roots secrete acids strong enough to break down the bone's mineral structure, unlocking the fats and proteins trapped inside. The worm has no mouth, no gut, no anus, so symbiotic bacteria living within the roots do the rest, extracting the released nutrients and converting them into compounds the worm can absorb. No mouth, no gut. Just chemistry, partnership, and patience, sustaining life in one of the most barren places on Earth. There are an estimated 690,000 whale carcasses on the seafloor at any given moment, each one a temporary oasis. Food is a problem life solved, ingeniously, repeatedly, at every depth. The next challenge is colder. Which of these do you find most fascinating? The vampire squid, the zombie worm, or something else entirely? Drop it in the comments. Temperature in the deep sea is always on the verge of freezing. In the Southern Ocean, around Antarctica, it drops below zero. Life's response, counterintuitively, is to grow bigger. Most sea spider species are a few millimeters wide. In Antarctic waters, they reach 500 times the size of relatives elsewhere. The Greenland shark grows up to 21 feet, and there are Greenland sharks patrolling the deep right now that were already old before Shakespeare was born, before the United States existed, and before the telephone was invented, making it the longest-lived vertebrate on Earth, and just one example of a pattern that repeats across the deep cold. Giant isopods, colossal squid, creatures that elsewhere would be small, here become enormous. This is polar gigantism. Cold water slows metabolism and increases cell size. It also holds far more dissolved oxygen than warm water. A larger body has more gill surface to extract it. In frigid water, being big is a breathing advantage. The crocodile icefish takes a different route entirely. It is the only vertebrate on Earth with no red blood cells, no hemoglobin, no color in its blood at all. Instead, its gills are proportionally enormous and its skin is completely scaleless. Both surfaces act as oxygen-absorbing membranes. The whole animal breathes through its skin. In the oxygen-rich waters of the Antarctic, that is enough. But what happens when the water drops cold enough to threaten the blood itself? In parts of the Southern Ocean, temperatures reach 29 degrees Fahrenheit. Ice crystals forming inside living tissue would rupture cells instantly. And yet 90% of fish biomass in coastal Antarctic waters comes from a single family, the notothenioids. Their secret is antifreeze proteins. When an ice crystal begins to form, these molecules bind to its surface and stop it from growing. The crystal is blocked before it can cause damage. 3 structurally distinct types of antifreeze protein have evolved in polar fish, independently at least 4 times. Evolution doesn't reinvent a solution that often, unless it is absolutely essential. Cold, like food, turned out to be a solvable problem. The next one is stranger and more dangerous than it first appears. Scattered across the Mediterranean seafloor are underwater lakes. Ancient salt deposits have dissolved back into the water, creating brine 3 to 8 times saltier than the surrounding ocean. This water is so dense it doesn't mix with the sea above. It sits in hollows on the seafloor with its own surface and shoreline, a body of water within a body of water. At the edges, mussel beds thrive on chemosynthetic bacteria, but the lake itself is something else. Across its floor lie perfectly preserved animal corpses, decades of casualties, some perhaps centuries old, pickled by the salt. This is a graveyard. To understand why, you need to understand what salt does to a living cell. Every cell maintains a careful internal balance, a precise concentration of water, salts, and proteins. Expose that balance to a hypersaline environment and it collapses almost instantly. Osmosis pulls water out of the cell faster than it can be replaced. Proteins denature, enzymes fail, and the machinery of life simply stops. For most animals, crossing into a brine lake isn't just uncomfortable, it is chemically catastrophic. The cutthroat eel learns this the hard way. It hunts the seafloor by smell, and one day, that nose leads it to the mussel-covered edge of a brine lake. It crosses in. The consequences arrive immediately. No oxygen, and a salinity that starts stripping water from its cells at a lethal rate. Its muscles lock. It writhes and convulses. The only way out is a fast retreat. And even then, it's close. Most animals that fall in never come back out. Except one. The Gulf hagfish is the only vertebrate known to enter a brine lake and leave unharmed. It has prowled the seafloor for 300 million years, and its secret is slime. When threatened, specialized glands fire within milliseconds, releasing close to a liter of material in seconds. The slime consists of two components, tightly coiled protein threads and tiny mucus packets. The moment they hit seawater, the threads unravel. The packets burst, and together they trap enormous amounts of water, expanding to 10,000 times their original volume almost instantly. The result is a gel thick enough to clog a predator's gills. Around brine lakes, this slime serves a different purpose. It coats the hagfish's body and reduces water exchange with the toxic environment around it, a physical barrier against the salt. Combined with an unusual tolerance for oxygen-depleted conditions, these two traits let the hagfish dart in, feed, and leave, where everything else stays. Salt had a solution too. But the final challenge is different in kind. It doesn't just push life to its limits. At a certain depth, it makes survival chemically impossible. And the reason is written into the very molecule that makes deep life possible in the first place. If you've made it this far, you're probably going to want to see how this ends. Subscribe, and let's go deeper. Below nearly 4 miles lies the Hadal Zone, named after the Greek underworld. It is 18 times deeper than the record for the deepest scuba dive. The pressure is 600 times greater than at the surface. Every air-filled cavity in a human body would collapse. And there are still 3 miles of ocean between here and the floor of the Mariana Trench. At the very bottom, pressure exceeds 1,000 times that of the surface. Temperatures sit just above freezing. No light, no photosynthesis. By any reasonable biological measure, vertebrate life should not exist here. And yet, the Hadal Zone is not empty. The big fin squid is one example. This rare, spider-like creature drifts through the Atlantic and Pacific at nearly 4 miles down on soft, gelatinous tissue. It has no rigid skeleton and no gas-filled spaces. No swim bladder. Nothing that could crack or implode under load. Instead, a flexible internal structure called a gladius anchors its muscles just enough to function. Its body is mostly water. Under pressure, it yields slightly rather than shattering. The Dumbo octopus, found at nearly 4.5 miles down, has arrived at the same conclusion. It is the deepest known octopus, and it has traded strength for flexibility. Low muscle density, a soft body, and a beak too weak to tear. It swallows prey whole because tearing is simply not an option down here. The sea pig, a pale, tube-footed sea cucumber that grazes the abyssal mud, solves pressure differently, not through body shape but through chemistry. Its cell membranes carry unusually high concentrations of unsaturated fats which keep them fluid and functional even under crushing force. Where most cells would stiffen and fail, the sea pigs keep running. These 3 creatures survive pressure by becoming soft or by adjusting their chemistry. But the snailfish goes further than any of them. Filmed alive at just over 5 miles down in the Izu-Ogasawara Trench, the snailfish is the deepest fish ever recorded. Its survival depends on a molecule called trimethylamine oxide, TMAO. This molecule wedges itself into proteins and holds them in their correct shape, even as pressure tries to collapse them. Without it, the proteins behind digestion, respiration, and reproduction would fold and stop working. The deeper a fish lives, the more of it they carry. Researchers confirmed this precisely, measuring levels in fish from multiple depths, including 5 rare Hadal snailfish from nearly 4.5 miles down in the Mariana Trench. The pattern was clear and consistent: greater depth, higher concentrations. So the same molecule that protects fish from pressure slowly pushes their salt balance toward a point of no return. TMAO is inherently salty. The more a fish produces, the saltier its internal chemistry becomes. For most shallow water fish, this isn't a concern. They are already less salty than the ocean around them, and they manage that gradient by drinking seawater and actively pumping out excess salt. It's costly, but it works. As depth increases, rising TMAO pushes the fish's internal salt concentration upward toward equilibrium with the seawater outside. At equilibrium, there's no gradient and no problem. But go deeper still, and the gradient flips entirely. The fish becomes saltier than the ocean. Water now flows in rather than out. The fish would need to pump water out of its body rather than salt, the strategy used by freshwater fish, which live in water far less salty than themselves and produce large volumes of dilute urine to compensate. Deep-sea fish cannot follow this path for 2 reasons. First, pumping water out requires enormous continuous energy, and deep-sea fish run on the tightest energy budgets in the ocean. They simply cannot afford it. Second, producing high volumes of urine requires a bladder. A bladder is a hollow, fluid-filled cavity. At Hadal pressures, that cavity would be crushed. There is no room for it in the design. When the researchers extrapolated their TMAO data, a number emerged approximately 5 miles down. Below this depth, the salt chemistry becomes unmanageable. TMAO would have to accumulate to levels that push internal salinity past the point of no return, and there is no biological mechanism available to correct it. The molecule that makes survival at depth possible eventually makes survival impossible. The deepest fish ever observed was caught just over 5 miles down, a mere 440 feet below this theoretical ceiling. Given the uncertainties of deep-sea sampling, the match is remarkable. No fish has ever been found deeper. The chemistry explains exactly why. Below 5 miles, fish disappear. Not by accident. By chemistry, a hard limit encoded in the physics of salt and pressure that no vertebrate has found a way around. But the ocean doesn't end there, and neither does life. In the final stretch of the Mariana Trench, microbes and single-celled organisms thrive. A xenophyophore amoeba nearly 4 inches across was found at nearly 6.5 miles down. And just above the Challenger Deep, nearly 7 miles beneath the surface, 200 amphipods were recovered in a single 3-hour sample. Without fish to prey on them, they flourish at depths that would kill every vertebrate on the planet. Perhaps they carry different molecules than TMAO, alternatives that let them go where fish cannot. We don't yet know. What we do know is this: from hydrothermal vents to toxic brine lakes, from the abyssal plain to the deepest Hadal trenches, life found a way in. The zombie worm dissolved bone with acid. The hagfish armored itself in slime. The nototheniod ran antifreeze through its veins. Food, cold, salt. Life found a way around each of them, more or less. Pressure was different. It set a limit that chemistry itself could not cross. And yet, just below that line, in the dark, in the silence, under a weight of water that would crush anything with a spine, something is alive. We came here asking why the deepest quarter of the ocean has no fish. The answer wasn't darkness, cold, or pressure alone. It was a single molecule doing its job too well. If you want more deep dives into the science hiding in plain sight, hit subscribe. There is plenty more ocean left to explore.